Have you ever wondered how your body grows, heals cuts, or replaces old cells? The answer lies in a process called mitosis. Mitosis is how one cell divides to make two identical daughter cells, and it is essential for growth, repair, and everyday life. Today we'll break down what mitosis is, why it's important, and each stage of the process step by step. Before going into the actual phases of mitosis, let's go through the different vocabulary. Mitosis is a type of cell division that occurs in eukaryotic cells, such as plant cells and animal cells. Its main purpose is to create two genetically identical cells from one original cell. Each cell has the same DNA, the same number of chromosomes, the same functions as the parent cell. This is different from meiosis, which creates reproductive cells. Mitosis is all about maintenance and growth. Next, why is mitosis important? Well, it's important for three main reasons. Growth. As an organism grows, they need more cells. Repair. When you get a cut, mitosis helps replace damaged cells. Replacement. Old or worn-out cells like skin cells are constantly replaced through mitosis. Without mitosis, multicellular organisms could not survive. Here we have all the different stages a cell could be in before it divides or while it divides. This consists of G1, S phase, G2, which are part of interphase, mitosis, of course, and G0, which is a state where cells are not actively dividing or preparing to divide. During interphase, the cell grows during G1, and then the DNA is copied in S phase, and then finally, in G2, the cell will do the last preparations for division. By the end of interphase, each chromosome consists of two identical sister chromatids joined at a point called the centromere. Mitosis has four main stages, often remembered by the acronym PMAT. These stages are prophase, metaphase, anaphase, and telophase. Let's go through each one. In prophase, the cell starts getting ready to divide. During this stage, chromatin condenses into visible chromosomes. The nuclear membrane breaks down, spindle fibers begin to form, and the centrosomes move to opposite sides of the cell. This stage allows chromosomes to be moved accurately later on. In metaphase, chromosomes line up along the middle of the cell, also called the metaphase plate. Spindle fibers attach to the centromeres of each chromosome. This alignment is critical. If it doesn't happen correctly, the daughter cells may receive the wrong number of chromosomes. In anaphase, the sister chromatids are pulled apart. Well, what happens here? The centromeres split, the spindle fibers shorten, chromatids move to opposite end of the cells, and once they're separated, each chromatid is now considered its own chromosome. In telophase, the cell begins to finish dividing. During telophase, the chromosomes uncoil back into chromatin, new nuclear membranes form around each set of chromosomes, the spindle fibers disappear, and at this point the cell has two complete nuclei. After mitosis is complete, the final step is cytokinesis. Cytokinesis is when the cytoplasm divides, creating two separate cells. In animal cells on the left, the cell membrane pinches inward. In plant cells on the right, a cell plate forms down the middle. Now two identical daughter cells exist. To summarize, mitosis is a carefully controlled process that allows cells to divide and create identical copies. The stages of mitosis are prophase, metaphase, anaphase, telophase, and they are followed by cytokinesis. Now, here's a short interpretation of what this process looks like in animal cells. Mitosis does not happen randomly. Rather, the cell cycle is carefully controlled by chemical signals inside the cell. Two of the most important control molecules are cyclins and cyclin-dependent kinases, also known as CDKs. What are cyclins? Cyclins are proteins whose levels rise and fall during the cell cycle. They act like timers, telling the cell when it's ready to move to the next stage. Different cyclins are made at different times, such as before DNA is copied and before mitosis begins. If the correct cyclin is not present, the cell cannot move forward. Next, what are CDKs? Cyclin-dependent kinases, or CDKs, are enzymes that help control the cell cycle. On their own, CDKs are inactive. When a cyclin binds to a CDK, the CDK becomes active and it sends chemical signals. It tells the cell to move to the next phase. Together, cyclins and CDKs act like a security system, making sure each step happens in the correct order. The cell cycle includes checkpoints, which are moments when the cell pauses to check for problems. There are 3 main checkpoints. The G1 checkpoint checks cell size and DNA damage. The G2 checkpoint ensures that DNA was copied correctly. And the metaphase checkpoint ensures chromosomes are properly attached to the spindle fibers. If a problem is detected, the cell cycle is paused, the cell tries to fix the damage, and if the damage is too severe, the cell may stop dividing completely. These checkpoints help protect the organism. Sometimes the control system fails. When a cell ignores checkpoints or does not respond to cyclins and kinases correctly, it can become a rogue cell. A rogue cell may divide when it shouldn't, divide too quickly, or divide with damaged DNA. This uncontrolled division can lead to cancer. Cancer begins when mutations affect genes that control the cell cycle. These mutations can keep cyclins permanently active, prevent CDKs from turning off, disable checkpoint proteins, and as a result, cells continue dividing even when DNA is damaged, the cell is crowded, or the signals say to stop dividing. Over time, these abnormal cells can form a tumor. Thanks for watching BioBonanza, and like and subscribe for more videos just like this.